Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We'd like to thank the Ann and Gordon Getty Foundation and Camilla and George Smith, whose generous support made this episode of Lunch Break Science possible. Here with us today is Leakey Foundation grant recipient Enquye Negash. Enku, thank you so much for being here with us.
Thank you very much for having me.
Enquye is joining us from Washington, D.C., where she is a postdoctoral scientist at the Center for the Advanced Study of Human Paleobiology at George Washington University. Her research focuses on understanding the ecological and environmental context of locations in East Africa where hominin fossils are found. This research has taken her to fossil sites in Ethiopia to analyze vertebrate fossils and fossil soils to determine what habitats were like millions of years ago. She's working with fossil collection. She's also been to locations in Ethiopia, Rwanda, and Mozambique to study modern ecosystems to better understand past environmental conditions. So here's just a couple of quick, teasers of some of the locations she's been, but we'll be looking more in depth at them in a very short amount of time. Today, she'll be discussing some of the projects she's working on to better define the habitats of our hominin ancestors. Before we hear from Enquye, though, if you are watching us live on YouTube, Facebook, Twitter, or Leakey Foundation Live, you can post questions for Enquye at any time in the chat, and she'll be responding to those questions live during the episode. The earlier you get those questions in, the more likely she will be able to answer them. My first question for you is, what types of environments were our hominin ancestors living in?
Well, that's a very broad question because when we say hominins, we're referring to our ancestors and close relatives in the last 6 million years or so. So depending on, for example, the time period that we're looking at, let's say if we're looking at the last 4 to 5 million years, or whether we're looking at the last 500,000 years or so, the environment is going to be very different. And also depending on different sites, the environment is going to be very different. If we look at modern ecosystems, for example, they have different composition of vegetation types. So in terms of hominins, our general understanding is that earlier hominins that had a significant component of arboreality or tree-dwelling behavior lived in more wooded settings compared to, let's say, members of the genus Homo that likely lived in more open or grassland-dominated settings. And generally, we think of this as mosaic habitats because it's not a complete forest or it's not a complete grasslands. What we usually find is mosaic environment, which basically means a mix of different habitat types or different environments.
So what methods do you use to determine the type of paleo environment?
So there are different approaches of studying paleo environment. If you're looking at a continental scale or a regional scale, you can, for example, look at ocean drill cores that give you a much more wider scale environmental information. But for hominin sites, what we usually do is look at the outcrops and study the materials that's within this fossil-bearing sediments. And we use what we call proxies to study paleo environments. And what these are basically materials that are found in the geologic or the fossiliferous sediments. And these proxies can be, for example, they can be physical proxies, such as, let's say, sediments or studying at the sedimentary structures. They can be biological proxies, which basically means remains of plants or animals. They can be macroscopic, they can be microscopic, but also they can be chemical proxies. which means by studying the chemical composition of this material, we get to say something about the environment. For my specific research project, I also use different types of proxies. I use biological proxies known as phytoliths, which are microscopic remains of plants. But I also use chemical proxies, such as studying the chemical, or more specifically, the carbon isotopic composition of, let's say, fossil teas or soil, to have a better understanding of the vegetation.
I love that you look at the different proxies and different methods to help you reconstruct. It's so cool. When did you become interested in pursuing a career in science?
Well, I have always been interested in science, but what sparked my interest in paleontology was when I took a course in high school biology, and when we were learning about evolution. That's when I learned that the fossil record is one way of studying different evolutionary processes, and that kind of stuck with me. So when I was doing my undergraduate degree in geology or in earth science, I took a course in paleontology, and yeah, I was fascinated by it. That led me to do a master's degree in paleontology. That's when I started studying about hominin paleoenvironments, and again, that led me to do a PhD in hominin paleobiology.
Yeah, what was that got you interested in focusing on paleoenvironments?
So my first experience of learning about hominins and hominin environments was when I was part of a field school in Malawi back in 2009. And for me, that was the first time learning about the hominin fossil record. And prior to that, I knew this was right around the time that I finished my bachelor degree and was planning to start a master's degree in paleontology. So even though I knew I wanted to pursue paleontology, I did not have a very clear idea which specific subdiscipline that I would focus on. So the field school was an eye-opening experience for me that made me narrow down my interest in paleontology. In the year after that, I went on to another field school in tectonics, climate, and human evolution. And this was back in Kenya and in Tanzania. And that also led me narrow down my research interest in hominin environments and hominin evolution in general.
So a lot of your work now is working to better define mosaic habitats. Why is this important?
Well, as I tried to mention in the beginning, most hominin environments are interpreted as being mosaics, which basically means a mix of different habitat types or vegetation types. And this is due to a number of reasons. And one reason can be because the fossil record by itself is very limited. Not everything on the landscape is preserved. And what we're dealing with is basically a time averaged signal of thousands of years, and in some cases, millions of years. So we're missing a lot of information because the fossil record is not complete. Another reason for reaching at these interpretations is that we use different proxies that tell us different aspects of the environment. But also, if we look at modern ecosystems, for example, mosaics are a very common feature of modern ecosystems. Unless we're looking at, let's say, dense tropical forests or this extensive grasslands, everything is a mosaic. What varies between the mosaic types is the proportion, the composition, as well as the configuration on the landscape. So that got me asking, can we better define mosaics in modern ecosystems? And can we use what we learn from the fossil record, what we learn from modern ecosystems to the fossil record, so that we come up with more refined paleoenvironmental reconstructions.
For this project focused on modern ecosystems, what were the locations you chose and why did you choose these specific locations?
Okay, so for this project that was focused on modern ecosystems, I focused on national parks because they represent a more or less undisturbed ecosystem. And I covered 9 national parks in Eastern and Southern Africa. One of the parks is shown here, Awash National Park in Ethiopia, is a park mainly dominated by, as you can see, shrublands.
That's gorgeous.
It is. Another park is Bale Mountains National Park, which consists of a range of vegetation types, like bush parks. Here, the alpine forests, or what's called an erica forest, is shown here. These are high altitude forests surrounding Bale Mountain National Parks, but the park generally has a range of habitats, ranging from the forests, the low and high altitude forests that's shown in this picture, but also high altitude grasslands and woodlands. Another park that I looked at is Mago National Park. And Mago is, as you can see, most of the landscape is dominated by bushlands. But again, it also consists of grasslands, patches of riverine forests, and high altitude woodlands. And in this specific picture, for example, you can see, even though it's bushland dominated, you can see a narrow band of gallery forest following a river. So like everything, it's a mix of things, but in some cases, one habitat type is dominant than the other. And here is Omo National Park. And as you can see that dense, very dense, actually, riverine forests that is found along the course of the Omo River. But again, in the Omo, we have this extensive grasslands, but also wooded grasslands, extensive bushlands. Again, a mix of different habitats or vegetation types. Another park that I looked at is Nechisar National Park. This is, again, dominated by grasslands and bushlands, but as you can see, this is a park that has been recently becoming more shrub encroached, or that is seeing a sign of woody encroachment. The other parks that I looked at are in Rwanda. This is Volcanoes National Park, and Volcanoes is very big. So my specific focus was looking at differences in vegetation types across altitude in Mount Misoki. Another park that I looked at is Nyungwe Forest National Park, again in Rwanda, and you can see this is mainly dominated by forests, primary and secondary forests. Another park that I looked at is Akagera National Park, again in Rwanda, which consists of a mix of bushlands and grasslands, as you can see nicely in this picture. And the last park that I studied was Gorongosa National Park in Mozambique, which is dominated by the acacia woodlands you see here, but mostly by Miombo woodlands. But also we have grasslands and patches of forests in this national park.
You can really see that there's this huge variety of what the ecosystems are like now. And it must have been so incredibly fun to get to go to these different locations.
It was. It was very exciting.
So Within the parks, how did you decide where to collect the samples?
So what I do in the first few days of the field is I try to drive around the parks to familiarize myself with the area. And in most cases, the parks assign me with an ecologist or a biologist that know the vegetation in the area very well. So in the first few days, I just do a survey to try to see what type of vegetation types are in the parks, but also to get multiple sampling locations. So I take GPS coordinates, and then after I have selected multiple sampling locations, I narrow it down by selecting the best sampling locations, and then I get to collect soil samples.
How common is it to find phytoliths in the soil?
So phytoliths
Or
are
at least in a soil sample?
so they are microscopic, right? So they are microscopic silica bodies that are found in plants, and they can also be found in the soil. And we will not be able to tell if phytoliths are preserved in the soil in the field. So we have to bring the samples back into the lab, and we have to do a series of extraction, which basically means separating the phytoliths from the rest of the soil. And then we get to study them under the microscope. That's when we know if they're preserved or not. But one thing I have to mention is, like I said, I'm focusing on modern surface soils. So if we compare fossil versus modern soils, generally modern soils have a higher chance of preserving phytoliths compared to fossil soils. But even in modern soils, it's going to depend on, for example, let's say the soil type, because even though they are considered durable because of their siliceous nature, sometimes they can be broken down, they can be dissolved, and especially if it's an alkaline soil, they can be dissolved. So to shortly answer your question, it depends on, if we're looking at modern soil, it will depend on the soil type, the soil pH, as well as the vegetation type, because while some plants produce abundant phytoliths, some plants do not produce that many. So it depends on different things.
So the work you're going to be talking about very shortly during your talk focuses on using diet to gain insight into ancient environments. How did you become connected with this project, and what was your role?
So this is a collaborative project that I worked in. And in the study, we were trying to study the diets of herbivores from the Shingura Formation. And to do that, we were looking at fossil tooth enamel collections from the Omo Turkana Basin. And this site is a hominin fossil-bearing site that is found in southern Ethiopia and northern Kenya. And the specimens that we were working on were actually collected in the 60s and in the 70s. So basically, we were working on museum collections. And how I became involved in that project is for my master's project, I did a project that studied the diet of fossil bovids or fossil antelopes in the Shangara Formation. So when the PIs of this large collaborative project decided to do a large-scale study of diet that is looking at the entire herbivore collection, I was invited to the project, and yeah, that was how I became involved. And my specific role for that project was basically to be part of the data collection process, starting from getting the powder enamel samples to the analysis, and also to the data analysis and publishing of the results.
I'm always curious, for both of your projects and projects like this in general, how many people are on the team that are working on them?
So for example, for the project that we were looking at, the diet of herbivores, it consisted of multiple people that participated and that helped out during the sampling process, but also during the laboratory analysis. But in terms of data analysis and publishing the results, it consists of people that you just saw in this picture. And for the other projects, one of the projects I work on is studying fossil soils in the Shingora Formation. And for that specific project, I work as part of the OMO Group research expedition, which consists of a team of paleontologists, geologists, and paleoecologists. In terms of my modern ecosystem work, so this is a picture showing the exposures of the Shingora during a field work. And for my work in Gorongosa, for example, I work as a part of the Paleoprimate project team. But for the other sites or for the other national parks that I look at, it's mostly me and a biologist or an ecologist that's assigned by the park for me, and one or two scouts. So it's the size of the team is much more smaller for the other national parks.
I'm really excited to jump into your talk now. Before we hear from Enquye, if you're watching us and enjoying this episode, please be sure to subscribe on YouTube and follow us on Facebook and Twitter to receive reminders of upcoming episodes. So now let's turn over the virtual floor to Enquye and hear more about her research. I can't imagine how cool it was to be able to go to all those different, just gorgeous locations. And those photographs were just so beautiful. Like I feel like, we have to edit and you can't include all the photos, but I think I could sit and listen to or watch them for such a long time. Okay. So yeah.
Yeah. So let me just quickly share my screen. Okay. Can Perfect. You see my slides?
Yes, I can. I will see you on the other side of your talk.
Okay. So in the next 10 minutes or so, I will try to talk briefly about my work on looking at woody cover in modern ecosystems and its implication for hominin environments. And as I tried to mention earlier, my main interest is looking at the paleoenvironmental context of hominin evolution. And one approach of doing that is by studying woody cover. And what I mean by that is basically refers to the amount and distribution of woody vegetation on the landscape. And a much more simpler way of understanding woody cover is if you imagine lying down on the ground, you can think of it as how much of the sky is what you see. Woody cover is a key ecological factor that determines the species abundance, distribution, the species type, and composition of the ecosystems. So for example, primates such as chimpanzees usually live in mostly in forests, while species like the wildebeest, they prefer open grasslands. So the composition of the fauna or the animals we find on the landscape is going to be determined by what type of vegetation is available on the landscape. Woody vegetation also has what we call multiple ecosystem services. So woody vegetation serves as a source of food for the animals. It provides shade. It's a source of tools, for example. It also provides, let's say, protection from different types of predators. And also it serves as nesting as it's shown in this picture by a sleeping primate. So when we talk about hominins, hominins, like every component of the fauna on the landscape, they benefited from the dietary resources, the shade, and the protection provided by woody vegetation. But woody vegetation cover is particularly important for hominins because it's tied to key adaptations in hominins, such as our upright walking or bipedal walking, running, endurance running, stone tool use, as well as this shade-seeking behavior. Therefore, to have a better understanding of our adaptations and human evolution in general, understanding the amount and distribution of woody vegetation cover on the landscape is very crucial. In order to do that, part of my work focused on studying woody cover in different types of mosaics in Eastern and Southern Africa. And when I was studying this vegetation structure, I was relying on proxies that can potentially be preserved in the fossil record. So in the sparks, I was collecting the soil, and I was studying the proxies that are found within the soil. One of the proxies that I used are the carbon isotopic composition of soil organic matter. And the basic principle behind using carbon isotopes to study the vegetation is that plants follow different photosynthetic pathways. C3 plants, which are usually trees, follow the C3 photosynthetic pathway, while C4 plants, which are grasses, follow the C4 photosynthetic pathway. Eventually, this leads to a very distinct carbon isotopic composition. And because the organic carbon in the soil is derived from the plants on the landscape, the carbon isotopic composition of the soil is going to be reflective of what type of vegetation was on the above ground. And in order to do that, I collect soil samples from different locations and bring the samples back into the lab. The samples are going to be dried, they are going to be weighed, and they're going to be measured. The isotopic composition specifically is going to be measured using an elemental analyzer mass spectrometer. And based on the results we get out of this analysis, we get to learn about the vegetation and the woody cover. Another proxy that I use are phytoliths, and basically phytoliths are siliceous bodies that are formed within the cells and tissues of plants. So when these plants die, these bodies are liberated into the soil, and they reflect. They also reflect vegetation type. And there are different approaches of using phytoliths, but the specific approach that I was using to study woody cover was using an index called tree cover density index, which basically looks at the proportion of morphotypes or the proportion of phytolith types that are indicative of woody vegetation to that of morphotypes or phytolith types that are indicative of grasses. So by looking at the proportion in this phytolith types, you can say something about the woody cover or the vegetation cover. And again, in order to do that, the samples will be brought back into the lab, and they will pass through an extraction process that involves using multiple chemicals to liberate or to extract just the phytoliths from the sediments or from the dirt. And then once the phytoliths are extracted, we get to mount them on a slide and study, count, and observe them under a microscope. Because of time, I would not go into detail about the results from this study, but generally what we have learned is that some proxies are better than others in distinguishing different woody cover types. But in general, we were able to come up with a statistical model that can predict woody cover based on these proxies. And this model can be applied into the fossil record to be able to make more refined woody cover reconstructions. To talk a little bit about my work in hominin-bearing sites, my study focuses on the Omo-Turkana Basin, which is a basin that is found in southern Ethiopia and northern Kenya. There are multiple sites or multiple formations within this basin. We have the Uzno, the Kibish, Mursi, Shangura, Nachukwi, and Kubifora. But my work specifically focuses on one of these sites, Shangura Formation. And this is a formation that covers the time span between about 3.5 to 1 million years. In terms of the hominin fossil record, hominins have been found from the Omo-Turkana Basin starting in the 1970s. And the Omo-Turkana Basin is actually one of the sites that has contributed significant amount of information about hominin evolution. Therefore, understanding the environmental context of these hominins is very critical in understanding human evolution in general. So to understand or to reconstruct the paleoenvironments, one of the proxies that I used are carbon isotopes into the enamel, and this is the collaborative project we did where we tried to look at the diet of fossil herbivores across time and trying to see how it's changed through time and trying to learn something about hominins. And similar to the soils, I've mentioned that C3 and C4 plants have different carbon isotopic composition, and herbivores that feed on plants, the carbon isotopic composition of the plants that they consume is going to be recorded in the body tissues of herbivores such as bones and teeth. So these tissues are going to record the information of the vegetation with some amount of fractionation. So by looking at or by studying the carbon isotopic composition of this enamel, we can say something about the diets, and by inference, something about the environment. And in order to do that, again, we have to take powder enamel samples from the tooth of herbivores. The samples will be pre-treated in the laboratory, and their isotopic composition will be measured by mass spectrometer. And then we get to analyze and study the results. And what we have found for our study is, so this is a results figure that shows the diet of herbivores in terms of the carbon isotopic composition for different herbivore types that we looked at. So we studied 9 mammalian families, including antelopes, primates, extinct herbivores such as the dinosaurs, elephants, equids, giraffes, hippos, rhinos, suids, and we were trying to determine how the diet of these herbivores was changing across time. So in this figure, the vertical axis is going to show you time, or it's labeled based on different members of the Shangura Formation, and the horizontal axis is is showing you a different isotopic composition. But to make it more simpler, the green shade is going to indicate a diet of browser, and the yellow shade is going to indicate a diet of a grazer. And generally, what we learn from the herbivores in the Shingura is that with time, there is an increasing component of C4 resource in their diets for most of the herbivores that we looked at. In terms of what that means for hominins, so in this figure, everything is combined. The results we get from individual values is combined. And we looked at more than 1,050 specimens for the study. And what we see is that there is a change in the distribution, a significant change in the distribution of the carbon isotope values at around 2.7 million years, and again at 2 million years. And what this basically means for hominins is, in terms of the Shingura formation, the hominin fossil record prior to 2.7 million years is dominated by a hominin species, Australopithecus. But the fossil record after 2.7 million years is where we have the appearance of the genus called Paranthropus, and also later in time, appearance of the genus Homo. So what this result is telling us is that since there is an increase in incorporation of C4 resources in the diet of herbivores, we interpret this as more woody vegetation being present prior to 2.7, but that after this time, there was an increasing in more open or grass-dominated landscapes. And with that, I would thank different organizations that provide permit, financial support, as well as laboratory facilities. And then I'll stop here.
Thank you so much, Enquye. That was a really fascinating kind of deeper dive into paleoenvironments. So we'll be taking questions from our viewers shortly, but I'd like to talk more about your skeletal remains conservation work, or I guess not talk more about it, but talk about it. So could you tell us about that project you're working on?
Okay, so that project I started thinking about that project back in 2012 when a colleague of mine, Zelalem Bedasso, and I were working on a project that was looking at the diet of herbivores. And we were traveling to different parks in Ethiopia, and we were trying to collect powder and amide samples. But what we found is that even though there is some collection, the number is not great, which limits your sample size. So improving this skeletal remain collections has been in my mind for quite a while. So when I had the chance to travel to Rwanda, I had the opportunity to take a training on skeletal remain conservation. And this is a project done in Rwanda with multiple institutions or multiple partners. So there I got to learn more about the protocols that are required for burial, excavation, cleaning, and curating the skeletal remains. So when I traveled to the different national parks in Ethiopia, I was trying to give short trainings for the park officials and the scouts on the importance of the skeletal remains, and also providing written protocols on how to do this type of work. And this is something I really would like to continue in the future. And in one of the pictures, I am working with my colleague, Chalacho Mastin, who has been actually doing an amazing job in the comparative anatomy collection at the National Museum of Ethiopia. And in this specific picture, this is a site called Kwasa, and we're trying to excavate remains of Theropithecus gelada. So and we are working with employees in the comparative anatomy collections in that specific picture. So he has been really working on the comparative anatomy collection at the National Museum. And my contribution, what I want my contribution to be, is to develop what's being done in the national parks, and also to create a better link between the museum and the national parks, so that we get to have this extensive collections that we can use for research, but also for educational purposes or for museum displays.
Yeah, Chalachu was on one of our lunch break science guests, so we've shared a link to his episode in the chat. He talks more about the collections during his episode, so definitely be sure to check that out. It's episode 3, in case you're not going to use the link and want to just Google it. So now let's take some questions from our audience. If you have not gotten your question into the chat, be sure to get it in as early as you can, because we have quite a few questions. So if you want your question to be asked, get it in now. Okay. So our first question is, let's see here. It comes from Patrick. How can this knowledge about ancient paleo environments help us with conservation?
Thank you very much for this question, Patrick. And this is a very interesting question, actually, because, for example, people who do climate modeling, the input they use for their models is basically what we gather from the fossil record, right? So understanding paleoenvironments has a very significant input in understanding future climate change. And in terms of conservation, again, this is a very good question, but this is part of a broader field, conservation paleobiology, where we get to apply the knowledge we learned about the fauna and the flora of the past in order to make better conservation decisions.
I think we have plenty of questions. Let's see here. What is our next one? Anne Marie asks, she says, good afternoon. And what do you use proxies in the same manner as indicator fossils for dating?
Indicator fossils for dating. Let me see if I understand this question. So if we're talking about using fossils for dating, we're talking about relative dating, which is relying on the fauna to make interpretations of different time periods. But the proxies that I specifically use are slightly different, because I'm not using it as a way of telling ages or dates, but rather I use the proxies in terms of what they can tell us about the vegetation, or what they can tell us about the environment in general.
Now let's take a look at our next question from our viewers. Also from Anne-Marie. Do you think there is a direct connection between large-scale environmental fluctuations and variability in human evolution?
Yes, there is. There is a connection between large-scale environmental fluctuations that are occurring globally, but we also have to be more cautious, because different areas respond differently. For example, if I talk about the Omo-Turkana Basin, even within a single depositional basin, at a specific time period, what you find in different sections of the environment at the different sections of the basin is going to be slightly different. So a more broader answer to your question, and would be it depends on large-scale fluctuations or changes in the global climate, but it's also going to be mediated by the local environments, or the local topography and vegetation too.
Let's take that. Okay. Lou asks, what depth do you sample the soil, A or B horizon, or simply a certain consistent depth?
Okay, thank you very much for this question. So for the modern soils, that I look at, I use surface soils or the A horizons, which basically would be the top 0 to 5 centimeters. But I also do work in fossil soils. And when I study fossil soils, I basically study pedogenic carbonates that usually form in the B horizon. So to simply answer your question, it depends whether I'm looking at modern soils versus fossil soils.
We'll take one more question from the audience. Do you think that understanding this can help us anticipate how landscapes might change in the future?
Yes, I think I tried to also cover this in the first question because if we have a very good understanding of what the modern landscape and what the modern climate is, and also if we have a good understanding of what the past looked like, it would make making, again, climate models or predictions in the future much easier.
So my last question for you is, do you have any advice for those watching who might be interested in pursuing paleoanthropology as a career?
So my advice would be take a course in one of the subjects one of the paleoanthropology courses that you think is interesting. If you have the opportunity, do an internship in a lab or in a museum. And also, if you have the opportunity, maybe going to a field school might be good. I think trying out different things to see if it is a right fit for you would be better.
Well, thank you so much for being here with us today, Enquye. It was a really fascinating opportunity to kind of, as I said, dive deeper into paleoenvironments. Next time on Lunch Break Science, we meet scientist Christopher Smith and learn about the development and life histories of primates, especially vervet monkeys. So again, just want to thank you so much, Enquye, for being here with us and being on Lunch Break Science.
Thank you so much for inviting me.
Yeah, it was really fun. And for our viewers, please stay tuned after the episode to hear more about Leakey Foundation programs. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge. Lunch Break Science is brought to you by the Leakey Foundation and is made possible by the generous support of the Anna Gordon Getty Foundation and Camilla and George Smith. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientist, and how you can help support human evolution research and educational programs like Lunch Break Science. Right now, all donations will be matched by generous donors, meaning your impact will be doubled. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming episodes and programs, as well as groundbreaking discoveries in human evolution research.